This IABS webinar on COVID-19 will start soon. Thank you for joining.
Welcome everybody to this second IABS webinar on the state of the art on COVID-19. Like during our May 27 webinar, we welcome very warmly the 4 speakers who did an excellent job at that time. Today they will give us an overview, each in their domain, about what we learned since May. And I have the feeling we learned a lot. The 4 speakers will each talk 15 minutes, one after each other. You are invited to pose your questions using the Q&A button on your screen. Your questions will be posed by myself individually or in group to the speakers. And if we have an overflow of questions, we apologize if all questions cannot be answered. Now I give the floor to the first speaker, Professor Arnaud Fontanet from Institut Pasteur, who will talk us about the latest data epidemiologically on the pandemic. Professor Fontanet, the floor is yours.
Thank you very much, Joris, and I'm very pleased to be here again and to have a chance to share with you some of the recent findings of studies we have performed, as well as discussing the epidemiological situation. As the title of my presentation shows, I will be focusing on Europe, but I believe many of the data I'm going to share with you will be also relevant for other countries, although we are at different phases of the epidemic, as you all know. So last time, I believe I shared that slide with you, which was a result of the work we had performed on mathematical modeling. The team of Simon Cauchemez at Institut Pasteur had been able to reconstruct the epidemic dynamics using data on hospitalization for COVID on the French territory. And they estimated, using mathematical modeling, that at the end of the lockdown period, which was mid-May for France, 5% of the population had been infected, with 10% for the 2 regions most hurt by the epidemic, which are the eastern part and the area around Paris, whereas the western part of the country was the least affected, Bretagne, and the rest of the country was in between, with an overall prevalence of the infection of around 5.3%, according to their estimates. The basic reproductive number had been estimated at 2.9, which means that for the virus to stop circulating spontaneously and creating epidemics, we would need to have at least 66% of the population infected before the virus would be circulating at the pace it is currently circulating. When
I'm sorry to interrupt, we don't see your slides.
I have to
Sorry.
send it again then, I guess. Let me try again. Does it work?
Yes.
Okay, sorry for that. So, well, you just see basically what I was describing to you, and a sort of a bit heterogeneous situation for France, which had to do with the initiation of the epidemic in the eastern part of France following an evangelical church meeting, and then the region of Paris, which had been very much affected probably by a cluster which started north of Paris and moved to the capital city. So again, I was mentioning that we need 2/3 of the French population to be immunized against the virus, whether it is through natural infection or vaccine, before the virus will start to be epidemic. One analysis we performed, which I'm sharing with you and has been published in the Lancet since, was trying to find out whether the lockdown had been efficient in France for stopping the virus circulation. And there had been another controversy, which was, why did we perform the lockdown at the national level, whereas it could have been sufficient to do it at the regional level in the eastern part of France and around Paris, since the epidemic was very active there, but not in the rest of the country. And you all understand that the lockdown had a major impact on the local economy. So what we did with Simon Cauchemez was to look at the hospital admission by region. And here you have the 13 regions of France. And the aim of this analysis was to show that first, it was truly the lockdown which stopped the epidemic wave. And two, each region was already in a dynamic that would have resulted in a major local epidemic had the lockdown not been performed. So to do this, we look at let's take this region, which is around Lyon. It is the second largest city of France in the middle eastern part of France. And what you see here is the epidemic curve of hospital admission. The scale here is number of hospital admissions per 100,000 inhabitants, and we use a threshold of 1 per 100,000 inhabitants for the hospitalization rate. If you convert that into number of cases every day on a daily basis, at the national level, that threshold would mean 150,000 infections. So you realize it means a lot of circulation of the virus. But at the beginning, before hospitalization, you don't see it. It's only number of new cases per day, but it really means the virus is already circulating a lot. And what you can see here on this slide, you have here the lockdown, which was 17th of March. And here you have the peak of hospital admission, which here is 28th of March. It's exactly 11 days after the lockdown. 11 days, it is a time between infection and hospitalization for those who do a severe form of infection. So in this region, it looks like indeed it is a lockdown that stopped the epidemic and dynamics, if you want, as it's exactly these 11 days that separate infection from hospital admission. The other interesting point is that you can see here that around March 15, 2 days before the lockdown, we already had reached a stage corresponding at national level, if we were using the same rate, to 150,000 new infections per day. So it's already extremely high. And therefore, here you can see that we had a very dynamic epidemic in this region of Lyon, and that the lockdown stopped it. And if you continue now throughout France, you will see that basically it's more or less 11 days after the lockdown that we had the peak, plus or minus 2 days for the hospital admissions. And you will also see that the threshold of 1 hospital admission per 100,000 inhabitants has been reached in all regions of France by the time of observation here, which means that when we compare the region where the dynamic has been the fastest, which was the eastern part, and this threshold had been reached on the 10th of March, and the region where the epidemic has been the slowest, which is Bretagne, western part of France, and the threshold has been reached on 23rd of March, all regions of France had an epidemic dynamic which would have resulted in a massive epidemic had the lockdown not been there. So that paper shows, 1, it is a lockdown that has been efficient. 2, with a shift of 13 days, all regions of France were all going towards a major sanitary crisis. So lockdown has been efficient and was necessary everywhere, and not only in the two regions which were the most affected, like Grand Est and Île-de-France. Now, if we look at what happened in the rest of Europe, you will see that similar to what we estimated for France, for most countries, including Spain, for instance, which was one of the most affected countries, the proportion of people infected, and these are surveys based on antibody testing, show that 5% of the population had been infected. 10% if you go to cities like Geneva or Paris, for instance. But Sweden, end of April, was 7%. But basically, all Europe was about at the same level, with, for the countries most affected by the epidemic, like Spain, Italy, France, 5% of the general population infected. And countries, maybe the UK a bit higher and other countries a bit lower. And for France, the serological survey found exactly the same finding than the one I was sharing with you through mathematical modeling. So it's really interesting to see that both ended up with the same estimate. But again, for Europe, we are very far from this herd immunity that we would need for the virus to stop circulating spontaneously. Now, what happened during the summer? Here is what we call a heat map, where we compare 4 departments close to Paris. So Paris is the lower right, and here you have 3 departments around Paris. So the sort of suburban areas around Paris. On this heat map, you have here the age categories, starting from 0 to 9 years of age and going up 10, 19, up to more than 90 years of age. And here you have the time, starting from, it's by week, and this is a week 18-24 May, going through the summer to 5-11 of October. So here we have 5 months with the summer in the middle. And in the colors that you can see here, the redder you are, the higher the incidence rate by age groups. And when it starts to be red like here, it means that we are above the threshold of circulation of the virus that we consider becomes really significant. So you can see that May and June was basically green, and that in August, in the second week of August, in the 20-29 years of age, the incident rate started to become into the red area. And throughout the summer, up to October, you can see that the incidence rate started to increase in all age groups, reaching the older age group of the more than 70 here, or the younger age group, in a very similar way in the 4 departments. And I have exactly the same map, heat map for Marseille, south of France, and you would find exactly the same for other European cities or American cities. And it's really interesting because it shows you that the epidemic started in the young age group of 20-29 and gradually over a few weeks reached other age categories. And at that time, August in France, we had a rise in number of cases, but we did not have a rise in hospitalization because those young people did not make severe forms of disease. But by September and October, when the older age group were reached, then people started to go to the hospital. And that has been the topic of a lot of controversies in France. And I don't know to which extent it has been the same for other European countries, for instance, or elsewhere in the world. But there were people saying during the summer, the virus has mutated and you do have an increasing number of cases, but the disease is much less severe than what it was in last March. We won't have a second wave of hospitalization. And all that was going on. The month of September for us has been hell, because in the mathematical models that the team of Simon Kochmelz developed, it was very clear that it was the age of the people infected that was explaining why we did not have hospitalization. But all those people in the media attracted a lot of attention, and people were very happy to hear that the virus had changed and that the disease would no longer be as severe as before. So we had very, very hard time to make people realize that unfortunately, the virus was back. And here I show you the modeling effort that was performed by the team of Simon, where you can see here is intensive care unit to the left and hospital admissions to the right. And this is an exponential curve. So we have here exactly the same rate of an R at 1.4. So it's a 40% increase per week because the time serial interval is 1 week. 40% increase in week. You don't see it at hospital ICU or hospital admission when you are at very low level. But if you continue exactly the same dynamic, at a certain stage, which here is the month of October, you see that the hospital admission in intensive care unit and conventional hospital becomes really high. And here is the threshold of number of beds we have for Paris. Unfortunately, during the month of September, when we were saying it's coming back, if it continues at that rate, we will saturate our intensive care units. Nobody was here to listen because people were saying, look, there are no one in the hospital beds and the virus has mutated and it's all over. And fortunately, we woke up too late with measures that you have seen in France, but other European countries have become serious. And this is going to be my last slide. I'm showing you here the dynamic of number of cases for several European countries. And what we have seen in France and might have been true for other European countries was the first wave that you see here for France, where we had the people coming back from holidays, going to school, universities, getting back their jobs, and in the cities, quite some tension, but somewhat after 3 weeks, we had the feeling that it was stabilizing. And the week of the 5th of October has been of a very different nature, with a very sharp increase in number of cases that we have seen also in all other countries, maybe not Spain at that stage, but the other ones, you can see that it's rising very high. And something has happened during the week of 5th of October in all European countries and all regions of France simultaneously. Beyond that effect of coming back to school, universities, and jobs, I wonder, but I don't have any data to document it, to support it, whether this was not the second effect that we were anticipating. When it becomes colder, people stay at home, confined environment, higher survival of the virus in colder climate, and the second effect. So we had, as the Scientific Council always said, by September, October, be careful. There will be people coming back to work, school, universities, and that's probably what we had here. And then it will become colder, and maybe, but I don't have yet the data to support it, then things become even more tense. And France, as many other European countries, in the last 7 days have taken some measure. For us, it's called a curfew. The term is a bit extreme, but we are just closing places at 9 in the evening, and people are no longer allowed to get out of their place except for professional or emergency reasons. So this is my update for where we are now. I'm sorry that the news are not good. And I hope that my colleagues will have better things to tell you, maybe around the vaccines with Melanie, I don't know. But now we have to be ready for a tough winter. And I think we all have to be extremely careful and use all what we have learned to fight this virus, because we will need it for the coming month. Thank you very much.
Thank you very much, Professor Fontanet. Now we pass over to the second speaker, Professor Bruno Lina from Lyon University. Professor Lina, the floor is yours.
Thank you very much, and thank you for this invitation to share with you some of the information. I'm sorry, I'm not sure I'm going to provide any very good data. One of the things I will provide to you is what we understand in terms of knowledge about the virus and how the virus is moving, or whether it is moving or not moving actually. So I just put my slides on the screen. Sorry about that. Should start from the beginning. So I want to go back to the different stage the virus went through during its emergence, and what is the evolution that we have seen in terms of evolution of the virological genome, and whether these evolution have had any impact in terms of transmission. So the virus, you have to remember from the very start that this virus is a respiratory virus, obviously, but it is from the beginning very much transmissible. And when you look at these viruses, as you know, the coronaviruses, they have all the same shape. They look like the one I can show you here. It's a round virus with these little dots here around the envelope of the virus, which is making this virus called the coronavirus, because it's like a crown that you have around the virus. And this virus actually, when we grow this on epithelial cells, human epithelial cells, we could see that it was hugely well replicating, and the system was very efficient. So this virus was really fully adapted to humans. But we also knew that it was a new virus coming somehow from a reservoir. And the most likely reservoir for coronavirus are bats. And you know that we have had two previous experiences of the introduction of viruses from bats, which is the SARS and the MERS. And these two viruses came from this reservoir, jumped into another species, which is the civet cat for SARS, and the dromedary for the camel, sorry, for the MERS. But even though these viruses went to another species and then subsequently went to the man, they were not fully adapted to humans, because the transmissibility of these viruses were not as good as what we have seen with the SARS-CoV-2, although there were some cases of transmission and some cases of very large transmission with the SARS. But still, we could get most easily control of these viruses by some very basic hygiene measure, things that doesn't happen with the SARS-CoV-2. And when we look at the very first genome that was available, that was a virus that has been collected in a case during the very early stage of the epidemic in the Wuhan wholesale market. Actually, this virus was really amongst the beta coronavirus, and it was very close to a bat virus that has been discovered a couple of months before. And the similarity between these two viruses was very high, more than 96%, as it is said here. And it was quite different as compared to other coronaviruses. So there was some kind of link, obvious link between this virus and this bat virus and the emerging virus. And when we look at when we were looking for the animal that should have been involved into the transmission to humans, you know, because the bat is not frequently transmitting directly to human, we had the first idea that there might be the pangolin involved into the transmission, because in the Guangdong area, which is not exactly near Wuhan, but still it's in China, there has been a couple of these coronaviruses quite close to the SARS-CoV-2 that have been detected in these animals. But when you look at the phylogenetic tree of these viruses, actually you have the bat one here, which is very close to the human ones that are in blue. And you could see that the pangolin viruses actually are much more diverse than was the bat virus, which tells us, when you look carefully to this phylogeny of these viruses, that there might have been an introduction from of this bat virus into the humans and the pangolin, but the connection between the pangolin virus and the human virus is really not obvious. So there might have been another animal involved into this transmission and the adaptation to humans, but it has not been identified as yet. And the thing is that when you look at the virus, when it was emerging, different viruses have been detected in China mostly, and we had the first French virus that was sequenced on the 8th of February. It was very close to the virus that was in the Wuhan market, these viruses were very similar. And what we did see from the very early stage is that these viruses that were identified were highly related, and the homology between all these viruses was very, very high. But still, at that time, we have the idea that this virus was highly transmissible. And this is due to the fact that actually, when you look at the bat virus, and you compare the bat virus with the previous SARS virus at the spike protein, and looking at this at the protein sequence of the spike protein, which is involved in the attachment of the virus to the receptor being here in blue, you could see that there's huge difference between the SARS and the beta coronavirus that have been introduced into humans. So this is really a new virus, and this is really showed very well with the spike protein sequence. And when you look at the sequence, the protein sequence of the human SARS-CoV-2 virus, and you compare that with the sequence of the same regions, the spike of the precursor, the prototype precursor that is in the bat, you can see that most of the structure of the protein is identical. There's only in the region which is supposed to interact with the human receptor, ACE2, that some changes have been observed. And these changes may not have been directly observed in the bat, because it would have prevented these viruses to grow in the bat cells. So one of the possibilities is that through recombination with another virus, the rat TG13 bat virus somehow acquired the capability of binding very well to ACE human receptor, and then subsequently was introduced in human, and then was able to be transmissible from human to human. And this allows the virus to replicate in the upper respiratory tract, which is the mandatory requirement for rapid transmission within humans. So we have not identified yet where and with which virus did this evolution of the rat TG13 virus so that it became a human virus with the capacity of being highly transmissible. Since the beginning of the epidemic, there has been an enormous amount of sequence data that has been generated, and this data is shared openly on the GIZ database, which is really an asset for the surveillance of the virus. And what we identified very early in March, and this was related to an evolution of the virus maybe at the end of February, is that we could identify 3 genogroups. And the 3 genogroups, the G here, S and V, were associated to specific substitutions that were occurred in different proteins. And the G protein here, the G group, actually is the one which is most interesting, because I will talk about this. And this virus is going to take over the other one. This is a substitution in the spike protein, and this substitution has somehow be overwhelming the other viruses, because when you look at what happens in less than 2 months' time, actually all the G virus became very much predominant, but it never completely displaced the other one. So the theory we had at that time is that maybe these viruses was having some kind of better fitness of the virus, and actually it was possible for them to have a higher transmissibility rate. But the data that we had very early on that was not really crystal clear, and there's still a work going on. But if you look at the sequence data that is available yesterday, this is the picture where you could see that the G group have been subdivided in 3 subgroups, G, GR, and GH. And we still see some of these other genogroups. They don't completely disappear. They're still circulating, and we can detect them in different places in the world. So although we can say that this G group with the subgroups are really overwhelming the rest of the sequence that have been identified, these other subgroups remain, and they have not disappeared yet. So when you look at what kind of substitution can be seen at the level of the protein which is interacting with the ACE receptor, there was this D614G substitution, which is close to the receptor binding, but not necessary. And there's another group of substitution that has been recently identified from July and up to recently now, which occurred in the regions close to the receptor binding. And there's one very interesting, which is the S477 with the different possibility, RY, RI, RN, where we say that these substitution may have an enhanced capacity, may induce an enhanced capacity to the binding of the receptor of the ACE2 receptor. This is ongoing study, but if you look at the overall evolution of the viruses, and I take the example of what we have done in France in terms of genomic surveillance of the SARS-CoV-2, what we see is that within the sequence that have been sequenced from the early stage of the epidemic, saying from March until June, there's a very limited evolution of the virus. And this is due to the fact that we know that the coronaviruses have a poor spreading system and it doesn't accept much of the sequence changes. There's a correction in the sequencing. So when we look at the molecular clock of the evolution of these viruses, we see that the change that occurs in this virus is the maximum of 2 to 3 nucleotides per month worldwide, which means in a genome whose sequence is 30,000 viruses, a very limited evolution capacity. And when you look at the different SNP positions where this mutation can occur, there's also a very limited number of SNP positions that can be impacted, less than 500, which means that this genome is highly stable. And when we hear about mutations, substitution, risk of evolution, you have to remember that this is a coronavirus. This is not an influenza virus, and it is highly stable. So I just want to make an additional picture of what is the impact actually in Europe based on the ECDC data of this virus. And what we can see is that there's almost no country that can avoid having the circulation of this virus, where the virus is not evenly distributed over Europe, but still we can see that there are very large hotspots, and France is one of these. So in summary, and this is my last, I have the acknowledgement slides after that, what we know is that the virus that emerged was actually perfectly fit when it emerged in December. And this was due to the fact that all the preliminary requirement for optimal dissemination in humans has been done when the virus was emerging. The genome is highly stable, as expected for the coronaviruses. However, we must keep in mind that there are possibilities for recombination with other coronaviruses, human ones, and deletion that may also impact the virus. The introduction of the D161.4G substitution in February that led to have a predominant virus, we are still working on the fact whether it is associated to an enhanced transmissibility. There's some very interesting work going on showing that maybe it was the case, but this is something that occurred in February. There's this recent emergence of the S477N substitution, and again, we are looking whether it has an increased affinity to the ACE2 receptor. And what I want to share with you also is that all what I can tell you now is based on the fact that there's a rapid sharing of the sequence data from the viruses. So if you don't do it, please share as much as possible the sequence data that you have. I will just acknowledge all the people that are working with me and providing the data that can be helpful for the discussion today. Thank you very much.
Thank you very much, Professor Lina, for this fantastic overview and this, as a virologist, I should say, very passionate work. And I am sure it will raise a lot of questions in relation with epidemiology and also in relation with protection and immunology. And so now we come to our third speaker, Professor Arnaud Marchand, who will look at it from an immunologic point of view and learn us what he learned since the previous webinar. Professor Marchand, please. We don't hear you, Professor Marchand.
Many apologies.
Okay, thank you.
So, thank you, Joris, and thank you for the invitation to speak to this large international audience today. So what I will be talking about are three topics that have been discussed extensively over the last months, and that's the pathogenesis of COVID-19, the role of innate and adaptive immune responses, the possible cross-reactivity between other coronaviruses, non-SARS-CoV-2 coronaviruses, and the role it could play in immunity to COVID-19, and then the immunity induced by SARS-CoV-2 infection itself, and the implications this would have for the coming months for those people who have been infected by the virus. So this is a lot to cover in 15 minutes, so I will touch upon a number of concepts, and I will not have the time to go into the details, but there will be references for the audience to go for if they are interested.
We don't see your slides, sorry.
You don't?
No, I can share them if you want.
Can you see them now?
Yes.
Oh, I thought.
Thank you.
Sorry about that. So this is the overview. Many apologies again. So this is the overview I just mentioned. It was recognized very early on that COVID-19 has, SARS-CoV-2 infection is a spectrum of clinical presentation, and that the critical forms, the severe forms of the disease, are associated with systemic and organ inflammation. We have high levels of cytokines, inflammatory cytokines in the peripheral blood, and we do see infiltration of tissues with inflammatory cells. So when we talked about this in May, this is the working model that we discussed. It was conceived that the immune effectors that are able to control viral infections, including respiratory viruses, macrophages, neutralizing antibodies, and T lymphocytes, that those effector functions do not perform the way they should in the context of severe disease, and what they undertake is what they go for is exacerbated responses. The myeloid cells, especially, but probably also the T lymphocytes, are hyperactivated, produce inflammatory cytokines, and that contribute to tissue damage and dysfunction. At that time, it was proposed that antibodies could have pathogenic role in the context of this disease, and I will come back to that in a few minutes. Since then, a number of studies have been conducted, essentially confirming this working model, but looking more details at the cellular level and molecular level at what's happening primarily in peripheral blood. And those studies have found increased levels of inflammatory cytokines correlating with disease severity, especially IL-6 in intensive care unit patients. These studies, these systems biology studies, have looked in an unbiased way at the total population of immune cells in the blood, looking at the single cell, the transcriptome of those cells, and analyzing diversity of immune cell populations. And among the findings of these studies, we see that type 1 interferon production is reduced in severely infected patients. And so this could contribute to disease severity because, as you know, type 1 interferons are critical for viral control. When we talked in May, we already knew that coronaviruses are actually good at interfering with type 1 interferon responses, preventing type 1 interferon production, and also interfering with type 1 interferon responses, the action of type 1 interferon on target cells. Since then, we've learned that SARS-CoV-2 is doing that very efficiently, and potentially even more efficiently than SARS-CoV-1 and MERS. So the working model we are facing, or we are using now, is that under normal circumstances, when you have type 1 interferon production, that these by epithelial cells, for instance, or dendritic cells, this type 1 interferon can control viral replication. But when this type 1 interferon production or response is defective, this allows viral replication, and that viral replication will induce monocyte recruitment and will stimulate cytokine production, inflammatory cytokine production, and thereby lead to disease. Two breakthrough studies that have been published recently in Science strongly support a role for type 1 interferon in preventing life-threatening severe COVID-19. In this first study, in-moron errors of type 1 interferon immunity, so genetic defects in type 1 interferon immunity, were detected in a small group of patients, but these are rare mutations in genes coding for proteins involved in type 1 interferon responses. Perhaps more intriguing and interesting is the discovery that about 10% of the patients with life-threatening COVID-19 have auto-antibodies against type 1 interferon. So antibodies that the patients themselves produce and that block the effect of type 1 interferon and thereby their capacity to control the virus. The interpretation of these data is that these patients had these autoantibodies before the infection. So it's not caused by the infection itself, but it was present before and potentially promoted. The production of these antibodies could have been promoted, stimulated by the infection. So producing type 1 interferon, responding to type 1 interferon is critical for protection against severe COVID-19. Another topic that has been discussed in terms of COVID-19 pathogenesis is the potential role of antibodies and their capacity to enhance either infection or disease. As you know, antibodies, when they bind viral particles, they can promote their phagocytosis by macrophages and their destruction. In some viral infections like dengue, these complexes can be internalized and favor macrophage infection, productive infection by macrophages. So this is seen in dengue. It's unlikely to happen with SARS-CoV-2 because macrophages cannot be productively infected by SARS-CoV-2. Another possibility of enhancement is enhancement of disease. And this would involve the activation of innate immunity, complement activation that would promote inflammatory cytokines. So the presence of antibodies would potentially exacerbate the inflammation itself. This is obviously not a black and white process. It's likely to be dependent on the balance between neutralization on one hand and the inflammatory properties of antibodies on the other hand. In this very elegant discussion in Nature, Anne Irving and colleagues discuss these possibilities, but actually make a strong statement about the fact that there is very little clinical evidence that disease enhancement happens in the presence of antibodies against SARS-CoV-2. So although it's something that we should bear in mind for future vaccine trials, for instance, the evidence today is not strong. And because antibody responses and the effector functions of antibodies are very complex, this complexity calls for broad, complete, multi-parametric assessment of antibody responses to SARS-CoV-2 in patients. And this is what these authors have done. They've looked extensively at antibodies and their effector functions in hospitalized patients. And what they found is that convalescent patients, so basically the patients who after hospitalization survive, have an antibody response that's biased against the spike protein, whereas the diseased patients had a response biased towards the nucleocapsid. So it seems to be important that, so first antibodies can be protective, we knew that, but it seems that a response biased towards the spike protein may be beneficial and prevent fatal outcome. So I will not dwell too much on T lymphocytes. This is what we saw in May. There was evidence that T lymphocytes in the context of severe infections are depleted and are dysfunctional with limited antiviral capacity. And actually this concept have been supported by the studies that have been done since. And if you are interested in those aspects, which are a bit complicated to discuss in a few minutes, I refer you to this very good review that was recently published in Nature Reviews. One important thing is that even if we understand better the pathogenesis of severe COVID-19, the relationship between the immune parameters and the risk factors that have been associated with fatal outcome remain incompletely understood. In this, I think, important study conducted in the Western Cape province in South Africa, the authors have looked at risk factor for COVID-19 death in a number of groups, either a large population or patients with COVID-19 whole COVID-19 cases or hospitalized cases. And what they found is like what was found in other countries, sex, age, diabetes, and other comorbidities were risk factors. What they found is that tuberculosis and HIV were also risk factor for fatal outcome of COVID-19. So this shows that depending on the population and the environment, different risk factors, in addition to the ones that have been well characterized, could be important, and that may involve immune parameters. The second point I wanted to discuss, and the two next points will be shorter, the second point I wanted to discuss is the role of exposure to non-SARS-CoV-2 coronaviruses, common endemic coronaviruses, and the induction of immunity to SARS-CoV-2 itself. A number of papers have shown that T-cell responses to SARS-CoV-2 peptides, SARS-CoV-2 antigens, can be detected in people who have not been exposed to the virus, either because they haven't had any sign of infection, or even people from whom samples were collected before the epidemic. And what these data, for instance, show that peptide pools or fragments of the N proteins can be recognized by cells from unexposed, uninfected people. So there is immunity to immunity to SARS-CoV-2 in unexposed people. What we know since is that these responses are boosted by SARS-CoV-2 infection, and that the T cells express an effector phenotype, so they could have protective role. The possibility that antibodies could be induced in a similar way, cross-reactive antibodies could be induced as well, is very much debated in the literature. There is no consensus yet, but I thought it would be interesting to share this preprint with you. These authors have looked at convalescent subjects as compared to naive subjects, so serum collected before the epidemic. And so what they see is that in parallel to SARS-CoV-2 specific antibodies, they see that convalescent patients, so patients who have been infected with SARS-CoV-2, also have increased responses to other coronaviruses, suggesting that some cross-reactivity could occur at least in some patients. So we may not all be equal to SARS-CoV-2 because of our previous contacts with other coronaviruses. So that this is what these data suggest. Another parameter, immune parameter, pre-existing infection that might be important is the state of the innate immune system and what is called trained immunity, the capacity of the immune system to respond to a broad range of pathogens. And also, specifically, the role that vaccines like BCG could have by promoting this trained immunity and these innate immune responses. The possibility that BCG could have such effect is supported by a recent publication by the group of Netia and colleagues. And this is an interim analysis of a randomized controlled trial in elderly patients looking at the incidence of viral infections and respiratory infections over a period of 1 year. And so what this interim analysis is showing is that BCG vaccinated elderly patients have a twofold decreased risk of viral infections, especially respiratory tract infection. And the reason why this interim analysis was conducted is because there was an interest in testing BCG in trying to protect against COVID-19. So they had questions about the safety of the approach and potential efficacy. And what this study shows is that BCG is safe in that population and suggests that it has a protective effect. And we are looking forward to the results of those trials, looking at the impact of BCG on the susceptibility to SARS-CoV-2 and COVID-19. The third element of this talk relates to the immunity that's induced by SARS-CoV-2 infection itself. How durable it is a major question which relates to the occurrence of these recurrent infections that have been diagnosed over the last weeks and months. So what this paper shows is that actually IgG to SARS-CoV-2 in convalescent patients persists over a period of at least 3 months. So we now have more time that we can examine it. And so these IgG indeed persist at high levels over the following months, 3-4 months after infection, longer than IgA and IgM. I think it's important to emphasize that this study was involved primarily cases of hospitalized patients, and we know, and the literature is telling us more and more that patients with mild symptoms or asymptomatic patients may have a shorter duration of immunity. I will finish shortly. Just a few more points I want to emphasize about this emerging field, which actually has yet to emerge, which relates to mucosal immunity. And I think it's very interesting to see that in the 80s, CHIM studies, so human challenge studies, were performed for coronaviruses, and these studies showed that IgA in nasal washing as well as serum antibody neutralizing antibody levels were correlates of protection against coronaviruses. So we are looking, maybe in a few words, we are looking at the presence of IgA in the nasal wash of convalescent patients in a large cohort in Belgium, and we do see high levels of IgA in the nose of these patients, high levels in the saliva, and no big difference between the two sites, suggesting that saliva could be an interesting site for sample collection. We also see that T lymphocytes are induced by natural infection. So overall, we have immunity that's induced by natural infection, but the big question is how protective is this immunity? And so I will quickly mention that we are conducting a trial, a study at this time in Belgium, looking at 2,000 subjects that we will be following over the winter. Half of them will be convalescent patients, and half uninfected controls, to see whether natural immunity is protective, and because we look at baseline immunity to SARS-CoV-2 and other coronaviruses to identify correlates of immunity. Maybe one word about a study I like a lot that we are completing at this stage, and that's the effective transfer of SARS-CoV-2 antibodies to the newborn. We compare antibody levels in cord blood and maternal blood, and what we see is that those mothers who acquired SARS-CoV-2 infection very late during the last part of pregnancy are actually able to transfer SARS-CoV-2 immunity to their baby with antibodies promoting phagocytosis, complement activation, and NK cell activation. So I will be concluding here. So no, just emphasizing that moms have actually beaten big pharma because they've been immunizing their children already in May 2020, so well before pharma. So as a conclusion, I think we see a clear central role for inflammatory response, deleterious role, and defective entrant pathway in severe COVID-19. Neutralizing antibodies likely contribute to control of disease and are a good marker for protection in future vaccine trials. We are learning about the other functions of antibodies, and we still need to learn more. There is cross-reactive immunity with other coronaviruses, and this could contribute to SARS-CoV-2, need for more studies on mucosal immunity and for protective studies of infection. Productive studies of infection-induced immunity to guide vaccine development. So I think that to conclude, we've seen tremendous progress in our understanding and knowledge about SARS-CoV-2 infection and COVID-19, but you may feel that this progress actually doesn't lead to a major difference in the way we are dealing with this epidemic, because actually, like in 1918 with the flu pandemic, what is proposed is largely wearing masks and social distancing. I think that we will see in the coming months, we can be hopeful that the knowledge we have gained will allow us to develop new tools, therapeutics, vaccines that will allow us to deal with this epidemic in a very different way. I think a big challenge will be to make sure that these tools are accessible to everyone on the planet. And for sure, 2021 will be a public health odyssey. Thank you very much.
Thank you very much, Professor Marchand, for the extremely dense presentation, which we could fill a whole day, I suppose. But this is a question for later. Now it's a very good transition to Dr. Mélanie Savy, who will tell us how and where we are with the vaccines. Mélanie, the floor is yours. And thank you for participating.
Thank you, Joris, and thank you for inviting me to give you an update of where we are since May. And I think it's fair to say there's been a huge amount of progress in terms of vaccine development over the last few months, and I will attempt to summarize that in 15 minutes. So if you move straight to the next slide, in this first slide is just to show you that the WHO has put together a target product profile of COVID-19 vaccines. This is the preferred profile. So ideally it would have 70% efficacy, would be a single dose. But there is also a sort of minimum target product profile with an understanding that everyone is moving forward with speed and and really looking at where could vaccines be beneficial for COVID-19 with a minimum criteria of 50% efficacy, and obviously allowing two-dose vaccines to be made, especially when we're thinking about a completely new disease that we are trying to protect people from. So the next slide. So this slide represents what we know in terms of globally in how many COVID-19 vaccines are under development currently. So we have actually managed to count through publications, through public announcements, that there are more than 320 vaccines currently being developed against SARS-CoV-2. Many of them are in the clinic, and I'll talk about those in more detail. But you can see here there was a very wide range of vaccine platforms, from the classic inactivated platforms, live attenuated platforms, through to the more new and innovative platforms such as the messenger RNA vaccines. So if you move to the next slide. And this is to show you really how much progress has been made. So we're now in October, just over 9 months since the virus was first sequenced and the sequence was made available. And this slide shows you the number of trials that are currently ongoing with the various candidates. So there are 39 candidates that have already reached clinical testing. Normally in vaccine development, this would take multiple years to get to a stage even of getting into phase 1. And also with that in mind, there are already 10 candidates that are in phase 3 clinical testing. So these really come into 5 different types of categories of vaccines, from viral vectors, which could be anything from the adenovirus type vectors, through to measles vectors and many others. I mentioned already RNA, which is obviously a newer and quite novel technology. Also DNA, protein-based vaccines with adjuvants, and the classic inactivated vaccines. You can see here that part of the work that we are doing within CEPI and COVAX, that there are 8 of these are under COVAX, which I will talk about a little bit later. Just to focus on some of the more advanced vaccines here. First of all, to say really mRNA has revolutionized vaccine development. So the very first candidate that got into the clinic was the Moderna mRNA candidate, and it was only 66 days between identification of the sequence and starting a phase 1 clinical trial. And it was, I think, for the first time, the fact that the manufacturing of the material for phase 1 was not the thing on the critical path for moving into the clinic. So with all of these 10 candidates already in phase 2, phase 3 clinical testing, there are some candidates that are now getting very close to first efficacy readouts. So Pfizer today, their RNA candidate, their CEO sent out a letter to explain how they're planning to move towards license. So there are 3 critical areas. The first is the efficacy readout, and they do expect to potentially have enough cases for efficacy readout by the end of this month. The second piece that is critical is safety. And the FDA have set some strict rules as to how much safety data is needed and the duration of follow-up for submitting for emergency use authorization, and they're planning to have enough data for that by the 3rd week in November. And then the 4th piece is clearly having the manufacturing well under control at the scale that you need for licensure, and that is expected to be in the same time frame, which does mean that it is possible before the end of the year that the first regulatory submission for emergency use authorization may be made. Equally, Moderna, who started also very fast, believe that they will have enough clinical data for the first efficacy readouts by the end of November. and AstraZeneca, through their efficacy trials that are ongoing in the UK, South Africa, and Brazil, anticipate having sufficient data for first emergency use authorization submission before the end of the year. So we have to remember these are submissions. They have to be reviewed by the regulators and approved for use. Next slide, please. So I want to then focus on, there's quite now a lot of clinical data available from the early clinical trials, and you'll see on the next slide really just a summary of some of the immunogenicity data that we're beginning to see from the candidates. So currently the candidates have really been tested in adults, and we're beginning to get some data in the elderly as well. And this is just an example of 3 different platforms that have presented data from their phase 1 or phase 1/2 clinical trials. You can see here the Moderna mRNA vaccine here looking at neutralizing antibody, Novavax with their protein and matrix M adjuvant, showing both binding and neutralizing antibody. And then the single dose vaccine from Janssen with the Ad26 vector, again, looking at ELISA and neutralizing data. And what you can see really quite consistently is that the vaccines that have been tested in the clinic do generate binding antibody responses to the spike protein, which is the antigen in all of these vaccines so far, either the full-length spike or the receptor binding domain. There is varying degrees of neutralizing antibody, but neutralizing antibody is being observed in these vaccines. And the cellular immune response is very much Th1 orientated. Some candidates are beginning to show data in the elderly population, and in some instances, as what is often seen with vaccines, the immune response in the elderly is somewhat less than in adults, although we're seeing some candidates with clear, good, strong data in an elderly population. Next slide, please. I don't want to go into any detail. This is really just to make a specific point in terms of our ability to compare the immune response across various different vaccine candidates. You can see here that this is looking at either binding antibody or neutralizing antibody across the various developers that have published data, that they're all using different antigens from different strains. They're using different assays, and they're using different substrates for the assay. So it is very difficult to compare the candidates at this point in time. But as you'll see on the next slide, this is something that we're involved in at CEPI, is really trying to look at having standardized and high quality assays available to all of the developers. So one of the first things, NIBSC are developing antibody reference material and antibody standards that can be used in assays. Also CEPI is developing a network of centralized laboratories that will allow for testing for humoral immune responses, both neutralizing antibody and binding antibody, as well as some cellular antibody assays. And this is currently for any developer who is in phase 1 or phase 1, phase 2, and up to the limit of the budget is free of charge. So anyone here who is a vaccine developer, we welcome the opportunity for you to test your samples through this centralized laboratory network. Next slide. I think this just shows you the network that we have. And on this next slide, really just wanted to summarize what we're seeing so far in the clinic from a safety perspective. So overall, we've seen the reactogenicity profile is generally satisfactory. In some cases, you're seeing increased reactogenicity in the second dose. And there's been no clinically relevant safety signals to date. We do know that several trials have been paused to evaluate safety. This is quite normal in clinical development, that if you see an event close to vaccinations that may require hospitalization, you pause while you look at that. We have not seen evidence so far of vaccine-mediated disease enhancement. and we need to think in the long term of continuing to gather safety data. Next slide, please. I won't focus too much on the next couple of slides, but from a efficacy perspective, we now have multiple efficacy trial designs that have been published by the developers. You can see there are some nuances and differences here. In terms of the efficacy trials that are being conducted, they are in adults and the elderly. And you'll see on the next slide that the statistical assumptions and sample sizes are sometimes different across the various studies, either aiming for 60% or 50% efficacy. And what has been really the agreed on lower bound of 30%. And you can see here the various assumptions, meaning that the trial designs are of different sizes, but really sizable number of individuals taking part in these efficacy trials. Next slide, please. So then moving on to a little bit on terms of partnership in the context of vaccine development. And in the next slide, this is something that's really come together. Well, probably came together at the time of the last meeting, but the Access to COVID-19 Tools Accelerator, which is led by WHO, is really looking at coordinating activities towards COVID-19 with a pillar on diagnostics, therapeutics, and vaccines. And for vaccines, CEPI is a co-lead of the initiative for vaccines, where CEPI is responsible for R&D and manufacturing, Gavi for procurement and delivery of vaccines, and WHO leading policy and allocation. So if you move to the next slide. So this is showing under the umbrella of COVAX what we are all trying to do. We have developed a largest portfolio of, largest and most diverse portfolio of vaccines. And some of those were represented in the earlier slide. So we have 9 vaccines in development, of which 8 are in clinical trials and 3 in late phase development. And the objective through a coordinated approach is to deliver 2 billion doses of vaccines by the end of 2021. And that those vaccines are distributed in a fair and equitable allocation process to all the countries around the world. Next slide, please. So where we are today, so COVAX has been formed with the portfolio of vaccines that we have, that we hope will deliver the vaccines by the end of 2021. And we now have 181 countries that have joined COVAX to have access to doses of vaccines for their population. And within that 181 countries, 92 of those are low and middle income countries for which there is fundraising for an advanced marketing commitment to ensure that the low and middle income countries have access to vaccines at the same time as the high income countries. So to avoid, you know, a small number of countries having access to the majority of the doses. Next slide, please. The last couple of slides really are going to focus on a little bit on manufacturing, a critical piece we're advancing in terms of clinical development, but we need to have the scale of manufacturing. And this is just to show you that there are a large number of commitments that have already been made for manufacturing, billions of doses from various manufacturers in bilateral deals, which means a huge amount of scale up of manufacturing, scale out, tech transfer, all around the world to look at potentially providing vaccines. This is in the context that we must realize that not every vaccine candidate is likely to succeed, either from a safety, efficacy, or a manufacturing perspective. So next slide. So this is just showing you that indeed the need of looking at a truly global scale in terms of manufacturing, and specifically for COVAX to deliver 2 billion doses of vaccine by the end of 2021, we need several successful candidates. So there's a lot of emphasis on finding the manufacturing capacity, both in terms of drug substance, so making the bulk material, and drug products, so filling the material into vials, labeling the material, and making it available. So a truly global effort. And we have conducted a survey which shows there is the capacity that is available currently to ensure that there could be 2 billion doses of vaccine made available. But the efforts that we'll need to get there are tremendous. Manufacturing vaccines is a highly complex activity. Next slide, please. So these are just my concluding thoughts. So since we last met in May, there's been a huge amount of progress being made. We have data, early clinical trial data from multiple candidates, which is promising. However, we still need to have the first efficacy readouts, but we can expect those now before the end of the year. And manufacturing is already occurring at scale, and tech transfer is beginning. But this is really still only part way through the journey. There are many challenges that we still have to face, especially in the low resource settings. So most of the vaccines that we have in development today are 2-dose vaccines, and we will have to find an efficient way to deliver those. The data currently is focusing on adults and elderly. There are other important populations that do need to have data on how well the vaccines will work in those situations. We have to think of cold chain requirements. I highlighted the mRNAs. These are new and novel technologies, and the cold chain requirements for some of these are actually ultra cold chain. Durability of the immune response and the protection of vaccines is, as of today, unknown and will need to be tested, and whether or not we need boosters. We need to put some effort into pharmacovigilance to ensure that we are monitoring the safety of the vaccines even after they are introduced. And the regulatory burden is huge. Just in COVAX, having 181 countries that need vaccines by the end of 2021 puts a huge burden on regulators for labeling. Is every country going to want to label in its own language, or will there be some pragmatism from the regulators, I think is an important point. And making sure that the infrastructure is available for delivery and introduction of the vaccine is another critical point. So I'll leave it at this. So as I say, huge amount of progress made, but still a long way to go. Thank you for your attention. I think you're on mute.
I was on mute. Thank you very much, Melanie, for this fantastic overview, which in 15 minutes is on itself a huge challenge to try to overview what has been done since the month of May. I want to stress together with you that this is really a new era. 5 years ago, it would not have been possible to achieve these results, thanks to the effort of thousands of people, not at least those who developed platform technologies and all those who had an open mind in order to put them forward in a quite pragmatic way. As promised, if the audience allows me, as promised to Melanie, because she has to leave before, I will first pose the questions that concern the vaccines. So we have a couple of questions on the vaccines. So first of all, about what is your opinion about a minimum follow-up 2 months before an authorization? Secondly, we have questions on the target product profile. So the WHO target product profile, it's envisaging all ages. So do the manufacturers foresee particular emphasis on the different ages? And then there are 2 questions concerning the strains. So the question is, if there is a strain change or a modification, are the vaccines or will the vaccines sufficiently cover the different changes that could be expected or that could happen or not happen with the virus. Up to you, Melanie.
Great, thank you. So some very good questions, very pertinent questions there. First of all, on the 2 months. So indeed, FDA have really put some minimum requirements from a perspective of safety, with at least half of the population have at least 2 months safety data, safety follow-up from the clinical trials. I think this is really prudent under the current circumstances, because both the safety and the efficacy of the vaccines does need to be carefully evaluated. We all know that it is in the time frame immediately after vaccination that you are likely to see safety issues coming from a specific vaccine. So I think it is prudent under these circumstances to have a minimum requirement, but it does not get away from the fact that safety needs to be continued to be monitored, even post-emergency authorization. So longer term follow-up to understand if there are any safety issues in the longer term, and also follow-up as the vaccine is more widely used through pharmacovigilance systems and specific safety studies to evaluate what could be much rarer events that can sometimes occur following vaccination. So just to say, even once we get vaccines through to a first emergency use authorization, there's still a lot of work to do, and we should be carefully monitoring safety. In relation to the question on the target product profile, indeed, the ideal situation would be to have vaccination open to all populations. What we do know is that certain populations are specifically at risk of severe disease and even death. And so obviously the priority for the first evaluation is those types of populations. And as we know that vaccine manufacturing is challenging, there will be, even with COVAX's emphasis on 2 billion doses by the end of 2021, there will be a vaccine shortage. So the priority should be to vaccinate those most at risk of severe disease in the first instance. And those first clinical trials are focusing certainly on the elderly to ensure that there is good, robust data there. What should come then after that is the other populations, such as studies in children, where we know that children get disease, but they don't get severe disease. So this will all be part of the clinical development plans for the manufacturers. Then I think the last question on strains, I think I can maybe share that a little bit with the other participants here, other speakers. So Bruno Linner spoke about the emergence of new strains that are a little bit different from the original Wuhan viruses. The work that's been done on those so far seems to suggest that there's not a huge antigenic difference there, and that the strains in the vaccines will cross-neutralize with the more recently emerging strains that are available. Obviously, continuing to monitor strains and strain variation will be critical, and to continue to evaluate whether or not that means that the vaccine strains will still potentially cross-neutralize and protect against new emerging strains. But as I say, perhaps some other panelists will have some views on that point as well.
Professor Linner, he's there, but he seems to have left, right?
Yes, he had to leave.
Professor Marchand, from an immunological point of view? Or Fontanet, from an epidemiological, immunological point of view?
Professor Fontanet has left as well.
Marchand?
Okay. Yeah, so I think that from an immunological perspective, one could conceive that there will be some tolerance to the variance between strains. And so the extent to which this tolerance will allow us to control the variants to this virus in the future. We don't know, but I think that the data that are available and the comments made by Dr. Lina suggest that we can be relatively confident that the immune response that will be induced with these antigens will allow control of future strains, of future variants.
Can we leave it there on that point? I have two remaining questions on vaccines, very short. So there is a question on pediatric formulations and another question, if it would come out that none of the vaccine candidates would reach more than 50% effectiveness, what then? Melanie.
Thank you. So in terms of pediatric formulations, indeed, once the developers start looking at the vaccine in children, I think it's always important to work out whether a pediatric formulation may be different, for example, from an adult formulation. So this has to be part of the clinical development plan to evaluate the vaccines in children for potential use and to look at, to see if a pediatric specific formulation is needed. To the question of 50% efficacy, obviously we would all like vaccines to be as efficacious as possible. But I think one of the things that we need to bear in mind is that not all, well, I think there are actually very few vaccines that we can say are 100% protective. And there will always be some breakthrough cases. So 50% efficacy would have a significant impact on disease and on the outbreak. One other thing that we often see, so I showed you some of the data on the trial designs for the phase 3, and the case definitions there are for relatively mild disease. What we often see in vaccines is that the efficacy might be 50% against mild disease, but much higher against severe disease. So we're hoping to really not just look at, you know, disease in general and mild disease, but to really evaluate what the efficacy might be against more severe disease. So that 50% efficacy endpoint could really have a significant impact on disease in relation to COVID-19. We would obviously like that to be higher, but often we do see that you see higher efficacy against severe disease.
And the very last question that just arrived on vaccines is the attributable impact of the vaccine at population level and understanding of the commitment non-pharmaceutical interventions at the same time. So how could CP, WHO and other national or international authorities encourage sharing of data and summary mobility data, et cetera, et cetera?
Right, looking at the population level, I think this is going to be critically important, and I think there will need to be some trials done once the vaccines are introduced. Classically, pre-licensure, you're really looking at the individual level, but modeling can be done to see what the impact may be at a population level. And I think here, you know, part of this question is what level of vaccination do you need to get to herd immunity? This is a respiratory virus, and what we have seen from the animal data is that vaccines are preventing disease in the lung, but you do still see some virus shed from the upper respiratory tract. So there are two elements. There's direct effect of vaccines on preventing disease in the individual, but as part of looking at herd immunity, potential for transmission of virus is another important area. So we would imagine that transmission would be reduced, but it may not be completely eliminated. So in that regard, it's quite a complex story. And I think it's good to do some modeling to look at how various vaccines could impact at the population level, and to start looking at things like where herd immunity could be kicking in. There was a bit more to the question, but I'm not sure that I've answered all of it.
I think yes.
Okay.
Thank you very much. So now we go more. Thank you very much, Melanie. So if you can stay, because there will still other questions that all will have at the limit also impact on vaccination. So we come a little bit more to the virus. First question that came is, what about a reportedly SARS-CoV-2 virus identified already in November 2019? Is that right or fake news? Then we have another question concerning the lower frequency and morbidity in developing countries, and a question to that, linked to that, is if there is less morbidity, mortality in developing countries, would the childhood overall vaccination with BCG play a role, question mark? And then the role of the infectious dose in severity of disease. Unfortunately, neither Professor Fontanet nor Lina are there. So you, Melanie, and Professor Marchand, can you take up Professor Marchand?
Yeah, thank you. Interesting questions. I'll take the second question about the lower incidence of severe disease in Africa, especially. So I think that the reasons why this is the case are still debated, but several possibilities have been proposed. I think one of the dominant ones is the age stratification and the age of the cases. And so, as we know that younger people suffer less severe disease, this has been proposed. I think it's reasonable to conceive that the overall lower mortality and incidence of severe disease could be related to that. I think that there may also be possibilities to think about the role for immune responses and the magnitude or the quality of the immune response. I think that what we see is that the state of the immune system at the time of infection is likely to play an important role. So I think it's possible that different environments stimulate the immune system at different levels or in different ways, and that this could contribute to the way the vaccine, the pathogen could be handled. But I think this is hypothetical, and we don't have any data to support that. But I think that's a reasonable possibility. Having said that, I wanted to emphasize, and I know my talk was quite dense, but I was feeling that to mention a few highlights may be relevant to the diversity of the audience, and I wanted to mention these data about the study that was conducted in South Africa, where it is proposed. I don't think it's confirmed yet. It's proposed that HIV and TB, active TB or previous TB, would be risk factors for COVID-19 related death. So I think that it's true that overall morbidity and mortality is lower in Africa. That's what the data tell us. But I think that things could be a bit more complicated than that, and that there would be risk groups in southern countries in Africa who would have actually a stronger risk of severe outcome. And the final part of the question relates to BCG.
Yes.
So the possibility that BCG could have an effect, I think, is interesting, and there is a lot of interest around that. I don't think we have an answer yet about this possibility, but I think again that's a possibility. Whether it does that in adults in other parts of the world, we should know hopefully soon. But again, I think that notion relates to what I was trying to say regarding the environment. And our immune system is plastic, and it's adapting at the innate level. It's adapting to its environment, and we are learning that innate immunity is really critical for the way we are handling this virus. And one thing that's completely not explored is what happens at the level of the mucosa. Because most of these studies are looking at the systemic level. I believe that what happens when the virus gets into your nose is critical and very largely unknown.
In that sense, there is also the question about the infectious dose. Does it play a role? And if it plays a role, to what extent? And could it play a role also in relation with age? Why apparently younger are less susceptible to disease, not to infection, than others, etc. Both of you?
Melanie, are you inspired by this?
Sorry?
Question? Are you inspired by this question? Please go ahead.
Oh my goodness! No, that is a difficult question to answer. I think hypothetically there may be some differences. Maybe to highlight some work that is ongoing that might help to address that. There is a movement to look at human challenge studies, where looking at that as a model to test vaccines or even therapeutics. And I think looking at the infectious dose in some of those studies may well give you some insight into that. Although, it's difficult to say. I'm not aware of data that can specifically support that hypothesis at the moment.
I agree with Melanie, and I like the link you made with the human challenge. I think that's, while we are considering such approaches, obviously the infectious dose, the type of virus, but also the amount of virus that would be administered if we go that way is obviously critical. I think maybe we could say that the relationship between infectious dose and severity of the disease, even if it's not documented for SARS-CoV-2, it has been observed and documented for other viral infections, such as measles or where secondary cases in the family, often of varicella, secondary cases in the family are more severe than the primary case, and this is considered to be, I think there are data supporting that, considered to be related to the fact that the infectious dose for the secondary case is higher. So I think that possibility is supported by existing data with other viruses, and maybe even if the data are not available yet, I think it's something to consider.
Okay, we continue to receive questions on vaccination and on immunology. You still have a little bit time, Melanie?
I will give you 5 minutes.
Okay, then first the immune question. So regarding the calculation of herd immunity, is it known which percentage really developed specific antibodies, and is there effect of pre-existing anti-corona heterologous antibodies? Secondly, what is the role of natural killer cells, and what about COVID patients that produce antibodies detected by a serologic test? Would it be possible for the patients that immunity is more a cellular one than humoral one? And what about cross immunity from coronaviruses from animals? There we come to the One Health. Do other beta coronaviruses as bovine or canine coronavirus play a role or not, or is something known about it? Up to Arnaud, and then I come to the vaccine questions.
Yeah, you may have to remind me some components of this multiple question along the way. But so to the question, do we know? No, first to the question related to other coronaviruses. I think that yes, we do have immunity to other coronaviruses, and as I tried to explain, the impact of such immunity on susceptibility to SARS-CoV-2 is unknown and is currently being addressed. So it's there. I think there is, from what we know about the immune system and the cross-reactivity of those cells and molecules, I think it's reasonable to assume that this will have an impact, which could be both ways, I guess. But I would tend to think that a protective effect is likely, and we need to test this hypothesis. What is the percentage of subjects who are infected and who do develop antibodies? I think that the data suggests that this is the majority. But we do see cases, for instance, patients who have COVID-19 typical symptoms such as anosmia. We do see some of these patients who remain actually PCR negative along the way and who do not seroconvert. So I think that there is evidence that absence of detectable seroconversion there is evidence for that. And I think we need data to see whether in such, to another component of the multiple question, we need to generate data to see how many of these subjects would develop T-cell responses, which would contribute to protection in the second stage. I think if you look at this backwards, among within the studies where T-cell responses were examined following mild or asymptomatic infection, the authors have noticed that at the time of analysis, no antibodies were detected. So it's possible to have T-cell response at a given time without having detectable antibodies. So whether those patients didn't seroconvert at all, or whether they had transient antibody responses, is obviously very difficult to answer in a cross-sectional way. But the simple answer to that question is that I think that there will be variability in both antibodies and T-cell response, and that in part of the population, antibodies may be at low levels and or is at low level, and in those patients, T-cell responses could play a role. But all these are hypotheses that again need to be explored at in prospective studies at the population level.
There are some other questions coming on virology and immunology. I come back to you, but first the two questions we received on vaccines before Melanie leaves. And thank you, by the way, Melanie, for all your precious time. So first of all, there is a remark. There is a lot of talk about virus mutation, but finally, science shows that the virus is in fact quite stable. So, which for the vaccine development is rather a good news, I should say. And there is an important question concerning the post-marketing follow-up of efficacy and safety of the vaccines. How will this be organized in order that it's done in a sensible way, in such a way that things are compared which can be comparable, otherwise said, peers are compared with peers and apples with apples?
Yeah, so that's a very good question. So, as I alluded to, we have the efficacy trials which will get to licensure, but there will need to be both post-licensure safety and efficacy studies. That is an important part of the risk management plan, is to plan for those studies in the future that are needed from a regulatory perspective, but also obviously to understand in the real world setting how a vaccine, how effective a vaccine is. So this is an area that I think could benefit from some coordination, and this is certainly a topic that we're looking at with our partners at both Gavi and at WHO as to how to address the monitoring of vaccines post-licensure, but absolutely needed. And I think it's critical to have those elements in the risk management plan.
Thank you very much, Melanie. So there is another question, but you can probably answer quite short, in a short way. It's about the safety, particularly on mRNA vaccines. Mm-hmm. So there seem to be tolerability issues. So what is your experience about that?
Yes, so in terms of the mRNA vaccines, we have quite a bit of data now on the tolerability of those vaccines. They can be somewhat reactogenic. So you do see that there are local and systemic reactions in the days that follow vaccination. But what we have seen from the candidates that have presented data so far from Pfizer, from Moderna, is that those, they may be dose limiting. So part of the way of choosing the dose is based both on optimizing the immune response, but also on the safety perspective. But from what we've seen from those candidates is that, you know, the safety profile is in the same ballpark as many other vaccines in terms of the reactogenicity.
Thank you very much, Melanie.
Thank you.
Thank you. So we go to the remaining questions. Arno, another question on antibody production. Do all COVID patients produce antibodies, or at least detectable antibodies? And would it be possible for patients? So I posed the question already. So to come back to that, to be sure that we answer that question. And a second immunological question is concerning the BCG vaccination. So regarding the hypothesis of protection provided by previous BCG vaccination, how can we explain that the elderly population, which has been still extensively vaccinated with BCG, in contrast with younger population, do not, which are not much vaccinated with BCG, are in fact more at risk than the younger population? So what should we think about it in terms of a trained or non-trained immunity?
Thank you. So I think that the first question we already addressed. I think that the answer is that we have the vast majority of the patients presumably who develop antibodies. How long they last when they are at low levels is currently being examined. But there is certainly a fraction of the population that we see on a daily basis that has no detectable antibodies after some time post-infection. And in those patients, at least in some of them, we do detect T-cell response, which may be protective. So that was the answer to that question. The second question is interesting. I think it's important to realize that this is a multifactorial disease, and that the susceptibility will be the consequence of the integration of multiple parameters. Obviously, we cannot attribute the susceptibility of older patients to the disease to BCG or no BCG. BCG will not be the only or the main determinant of susceptibility to infection. What the observation that the person raises or mentions indicates that older age is associated with factors, determinants completely outside BCG, and that will impact disease outcome. So I think that the question that we want to address is slightly different. It is whether, in the context of the susceptibility of older patients to severe disease, can we change that with BCG? Can we change that by changing the set point at which the immune system is at the time of infection? And that's an open question. But obviously, we would not relate the susceptibility of the overall susceptibility of elderly patients to BCG vaccination or no BCG vaccination. It's more complex than that.
Okay, thank you very much. So another question to you. Could you address imprinting issues, so immune imprinting issues by vaccines in comparison or in contrast with wild type infection?
Okay, this is an intriguing question. I guess that the person refers to imprinting like lasting effect of an infection on the immune system at large. Yeah, I think that's an interesting question. So I think we don't know whether the natural infection will have long-lasting imprinting effects. We do see major alterations of the immune system during the course and soon after infection. How long will last these alterations? We don't know yet. But again, this is something that's being examined, and actually that's being examined at the clinical level in the study I was mentioning that we have initiated in Belgium, where we are following up convalescent patients and controls over the winter season. We will not only diagnose SARS-CoV-2 infections, but also other infections caused by other respiratory viruses, other coronaviruses, RSV, flu, etc. And the question, which is one of the secondary objectives of this study, the question is whether SARS-CoV-2 infection previous SARS-CoV-2 infection changes the susceptibility to infection by other viruses. This would, if that's the case, this would be a kind of imprinting. There would be confounders. Obviously, we would be looking at that. So I think that we don't know whether natural infection has long-lasting imprinting on the immune system or not. I think it's likely and it deserves attention, but I don't think we have the data yet. So whether vaccination will do something similar, I think is less known, obviously, because we've just embarked on those trials. I think it's less likely because, you know, as I said, the infection is generating massive alterations in the immune system, and I think it's very unlikely that even if a vaccine would be life attenuated, that similar changes would occur. So I would think that if major imprinting on the immune system is induced by natural infection, vaccines would have less of such effects. And if those effects are deleterious, I think that's one way to look at it as well. If natural infection has negative imprinting on the immune system, increases susceptibility to other infections, that would be a benefit of the vaccination, because the vaccine would prevent an infection that would have a negative effect. So indirectly benefit health through that prevention of imprinting. But all these things are very interesting. We don't have much information yet. But these are important questions that will be addressed in the coming clinical studies.
No, we received two similar questions. The first question is on the role of human challenge in testing of vaccines, but unfortunately, I think Melanie left already. But probably Professor Niels can answer that. And secondly, how to realize a study on efficacy of vaccines on nasal disease or asymptomatic disease, and particularly transmission of the virus. So these are two questions who are linked.
Yep. So Peter may want to complement, but I think people are right in thinking carefully about human challenge. I think it would be a major undertaking. It would be a serious decision to go for that. That can only be justified by an extreme need to generate the information that human challenge studies only human challenge studies can provide in a rapid way. But I think that depends a bit on how things will evolve during the coming months. Yeah. But indeed, some vaccines may not have, we talked about that, may not have the efficacy, the level of efficacy that we would like to see. There may be variability in the efficacy of vaccine candidates. And I think human challenge studies, carefully conducted, well designed, and you know, with all aspects of protection of the participants really under control, human challenge studies may be the only way to answer some specific questions regarding whether some vaccines are better than others, and what could be correlates of protection on which we could perhaps then build better vaccines. I think the need for human challenge studies will be better assessed in the coming months, but I think it is important to start thinking about them now in order to be ready if they are needed later.
Yeah. Can I just add a few things in this? I fully agree with you, Arnaud, that we should start thinking about challenge trials today, and we have started very intensively, because you should know that to produce the challenge strain takes you between 6 and 9 months to have a fully acceptable CMC dossier for a challenge strain. So if you would like to do a challenge trial in December, then you need to start before the summer break your development. That's one thing. Secondly, people are quite reluctant to do challenge trials with COVID-19 as long as there is no good accepted rescue therapy. And although we have remdesivir, we have corticoids, it is still some people die, yeah, even in the young population, and we don't have the full risk factors for healthy volunteers to exclude them. Yeah? So there is a lot of work ongoing on challenge trials. I'm pretty sure that some of the companies will be ready early next year to do the challenge trials. And I think I'm in favor for these challenge trials, because the benefit risk of these things are positive. I mean, people are dying anyway from COVID-19 today. And this is a benefit risk. If you could come up with good data from a challenge trial, even with the bad news that somebody died in the placebo group, then still you could have saved a lot of lives due to the fact that you get data and speed up the licensure of these things. And I will stop here. Thank you.
Thank you, Peter. So I come to two remaining questions. Is there any more data about recovered patients? Can they be reinfected again? And if so, why is this so, Professor Marchand?
Thank you. Yeah, that's a very important question that I was trying to allude to in my talk. And I think that we do have now clear evidence that such recurrent infections do take place. Proof that we are dealing with true recurrent infections is not easy. We need to show that it's a different virus infecting the same hosts some time apart. But that evidence has been generated, and the number of cases, well-documented cases, is not very high. But the evidence, the proof principle is there. So how commonly does it happen? We don't know. And I think that, again, it is only through prospective studies, well-conducted prospective studies, that we'll be able to see how frequent this is and what could be the immune parameters that could underlie this susceptibility. So two additional sub-points on this. As we've discussed, and there were several questions around that, as we've discussed, some individuals have low levels or undetectable levels of antibodies post-infection, yet the magnitude of their T-cell response is not always well documented. So there are reasons to believe that susceptibility post-infection can be dependent on the magnitude and quality of the immune response. But we don't really know. But I think that finding out how much natural immunity can protect and what would be the incidence of recurrent infection is important. I think the other dimension to this is the possibility that recurrent infections would be more severe, would be exacerbated by pre-existing immunity, SARS-CoV-2-induced immunity, the antibody-dependent enhancement. Does this take place? I think that the data are reassuring, that because even if some cases, the well-documented cases, show more severe presentation after the second infection as compared to the first infection, I don't think that we are seeing clinical pictures resembling what we would expect with disease enhancement. So I think that yes, we can be susceptible to SARS-CoV-2 following a first infection. How frequently, we don't know. It's probably not that common, but we don't know exactly. And learning from those cases will tell us a lot about protective immunity.
Okay, thank you very much. Here, a last question. Is there any information regarding cross-protection between Bordetella pertussis and SARS-CoV-2?
Okay, so I believe that this question may relate to the notions that we alluded to with BCG. So there is some evidence that the life attenuated pertussis vaccine candidate that's under development may have non-specific effects like BCG has. So I don't think that there is any evidence that there would be cross protection or a non-specific effect of pertussis on SARS-CoV-2. But I guess the question may come from there. So there is evidence that some life-attenuated vaccines, pertussis vaccines, may have non-specific effects. This may be relevant, but I don't think we have any data to discuss that.
Okay. And then the last question is about vaccines for animals. So the question is, if a vaccine manufacturers are trying to include companion animals in the development of vaccines. As far as I know, I think I can answer that. I did not hear that anyone is developing vaccines for SARS-CoV-2 in animals or companion animals.
And the justification for that is not so clear.
Well, there are only very few cases, and fortunately, I should say, it seems that the number of cases in animals is very limited. So I think we do not have any questions anymore. I would say if the speakers have any comment, but you are the only one left.
Only one left, can you see?
If you
it was a pleasure to be with you this afternoon.
Okay, and I would like to thank you, Arnaud, and the three other speakers for their very informative information. As usual, IABS will again make an overview of this webinar. Everything has been recorded, and this will be published in the journal, peer-reviewed journal, Biologicals. So thank you very much. Thanks to all the participants from all over the world for having participated and having posed so many interesting questions. And you will be informed about the publication, of course, and you will be informed about any new initiative of IABS concerning COVID or other important health issues that can contribute to a better global public health. Thank you very much. I will close here and hope to see all of you very soon, and I wish you a nice weekend. Bye-bye.